Hey, science research. Okay, so we're going to take a look at this notes packet on protein biochemistry, and we will um, go through some of the notes just so that you guys are up to date on it. So I believe for both classes we built the models of the molymod proteins, and so we have the final model here, and you can see that you've got different parts to it. If you guys remember, amino acids have a couple of main parts. Then we have the amino group, which is the group that's circled here. Okay. Then we have the side chain, which is the R group, which is this pink sphere here. And then we have the carboxyl group, which is this carbon double bonded to an O with an OH. Okay. Oxygen's always red, carbon is black, nitrogen is blue. Okay. And so when we look at these, right, here's our carboxyl group. Okay. Um, and then this is our side chain. Now, when we look at proteins and amino acids exhibit a property called chiral. Being chiral means that they're the same amino acid, but they exist in two different forms. One is called the L form, and the other one is called the D form. Okay, they're mirror images of each other, so it's just the same as if you take your hands and you put your right hand and your left hand together. So these molecules are actually referred to as right-handed and left-handed. Left-handed is the L form, right-handed is the D form. Okay, and these are known as enantiomers. And enantiomers are basically different forms of the same molecule. Okay, and they're mirror images of each other. Why is this important? Two reasons. Number one is different forms of enantiomers can react differently. There are different categories of enantiomers which can be of the same molecule. For example, there are L form and D forms of glucose. And depending upon the forms you have, that determines whether you get cellulose or if you get glycogen based upon the bonding. Okay, cellulose is made up of L and D forms of glucose, whereas glycogen is just made up of one form, which I believe is the L form. That's not too much to worry about. So with chirality, if you look at this, you can see the differences, right? If you were to hold two amino acids in your hands, the left-handed form, you can kind of see the way it's set up with the side chain and the um, hydrogen, right? They flip locations, okay? Um, and basically, what this comes down to in terms of being important for us with proteins is that all amino acids are the L form. Why is that? That's just how life did it. There's no other reason for it. All life just happened to use the L form of amino acids. Perhaps it was the first form that of amino acid that developed. We don't even know. But what it does show is that there's common ancestry. The fact that all forms of life use the same amino acids. What's interesting is bacteria, they convert them, the L forms, into the D forms for their cell walls. So only bacteria use D amino acids in their cell walls. Okay. Now bonds. Okay, so you made your molymod, and we know that we've got all these bonds here. Okay. Um, and these are all covalent bonds. So all of these lines here represent covalent bonds, okay, which is the sharing of electrons. Okay. And so we know that the bonds put molecules together. They hold molecules together. They allow electron movement between elements. Okay. And we're really concerned with three types of bonds in proteins. Hydrogen bonds, which we talked about already, right? They're very weak and they're transient. And they're simply a force of attraction between charged components of atoms. Ionic bonds, which are very strong bonds, and it involves electrons being transferred from one atom to another. And then we have covalent bonds, which is the sharing of bonds, sharing of electrons. So here's what covalent bonds look like. Okay, so we know that when two atoms share electrons, we know that all sharing is not equal. And when electrons are not shared equally, we call it a polar covalent bond. This is the type of bond we see in water. Okay, when the sharing is equal, it's known as a non-polar covalent bond, and the charges are distributed evenly among the two molecules. Okay, so a quick picture here of what these look like. In a covalent bond that's non-polar, you can see the little electron cloud here in blue. It's pretty well distributed evenly. Okay, you don't really see a high concentration of electrons on one end versus the other. However, when you go polar, You notice how there's a darker blue color here around the oxygen, right? And it's less intense around the two hydrogens. That shows us that the electrons tend to spend more of their time near the oxygen versus the hydrogen. And the result is you get a polar molecule. Okay, you have more negative charges hanging around this oxygen because the electrons like to go near the oxygen. Why? Because oxygen is more electronegative. So it pulls those electrons away from the hydrogens towards itself. Okay, hydrogen bonds, we know that in our water molecules, hydrogens of one water will always interact with the oxygens of another water. Some things that happen because of hydrogen bonding. We have adhesion and cohesion. Okay, adhesion is when different molecules stick to each other through hydrogen bonding. So I always remember this is an adhesive. When you want to stick something on the wall, right, you need to use adhesive tape. You need to use something to allow different things to adhere. Where cohesion is the same molecules and they are going to bond to each other. So that would be like water molecules interacting with each other, right? They cohere to each other, okay? Cohesion, perfect example of this, transpiration in plants, capillary action, which you've learned in earth science, and surface tension, okay? Interestingly enough, surface tension is exclusively cohesion, but capillary action and transpiration occur because of both the cohesive and adhesive effects of water, okay? And always remember that hydrogen bonds are weak, right? Individually, they're weak, and it's a weak interaction, but when you have many bonds together, it becomes very strong. It's kind of like Velcro. Velcro is the result of these little hooks interacting with the soft part of the Velcro. When only one or two of those interact, it's very weak. But if you get thousands of those little hooks to attach, now you've got a pretty strong bond. Okay? Right. And again, the hydrogen bond usually is indicated by dotted lines. And again, it's just showing you that it's this very weak force of attraction. But these are crucial to protein structure and stability. So if these two molecules come apart from each other, that hydrogen bond dissipates. That's why they're known as transient. Okay, ionic bonds. Um, if you have unequal valence electrons, this results in a strong charge difference. And literally, what happens, you can see with the little hand here, is one atom is going to pull the electron away from the other. So that makes the sodium become positively charged and the chloride becomes negatively charged. They become ionic. And so they form a very strong bond with each other and they form a strong crystalline lattice. And again, it's when one atom gives another atom its electrons. They often refer to ionic bonds as salt bridges. Okay. Van der Waals attractions are interesting because there's no real charge difference. But what happens is because electrons are always in motion, you sometimes get these um, fleeting moments where one end might be slightly positive and another end might be slightly negative. And so these molecules, they have to be really close to each other in order for the bonding to occur. So what tends to happen is nonpolar regions tend to interact with each other more often than not because of the slight difference that they get in the fluctuations of their positive and negative charges. And it's all because the electrons are constantly moving. Okay. How do they bond to each other? Amino acids. How do they bond? Well, they form a peptide bond, and we know it's through dehydration synthesis. So what we do is we remove an OH from one molecule and a hydrogen from another, and this results in water as a product. And so we get this bond between the two molecules. So where are the OH groups that get removed? One comes from the carboxyl group, and the other one comes from that hydrogen that's on the amino group. And when you do this, you create an open bond between this carbon right here and this nitrogen, and they form what's called a peptide bond. And it's because of the removal of this water. So here's that H and that OH, and here's a water molecule that they would form. Okay. So when we link them together, we can begin to form polypeptides. So if you take two amino acids and put them together with the removal of water, you now form a dipeptide, and that's what we have here. This is a dipeptide. It's two amino acids. Okay. Here's the OH of one group, and we take the hydrogen off of this nitrogen group, and there's our water coming out. And that carbon right here now forms a bond with that nitrogen, and you now have that peptide bond. So when you put these together, you can get a polypeptide. Okay. So take a look at this top diagram. By adding amino acids to your growing chain, you now form a long polypeptide. And the polypeptide, the key thing you're going to notice is these R groups. You see how they alternate? Up, down, up, down, up, down. Those are the side chains. The central portion here, you see you have these 2 carbons and a nitrogen, 2 carbons and a nitrogen. That forms what is known as the carbon backbone because it's just a repetitive sequence of carbons and a nitrogen, carbons and a nitrogen. The thing you'll also notice for a protein is that it has a direction to it. There is an amino end, which is called the N terminus, N for nitrogen. Okay, and then there's a carboxy end or carboxyl end, known as the C terminus. The reason why it's C terminus is that there's a carbon on the end, that's the carboxyl end. So this end is usually colored red because of the oxygens, and the amino terminus is colored blue because of the nitrogen. And that's the order in which a protein is read. It's always read from the amino end to the carboxyl end when you're looking at the amino acid sequences. Now, what makes amino acids different? It's the R side chains. It's those side chains, those R groups. So you can see in this bottom diagram that we now have our carbon backbone highlighted in gray, and these are those different side chains. And you can see some of these side chains, they're big, right? They've got this really big ring, like a tyrosine or a phenylalanine, and then some of them are small. Like a serine only has a carbon and an oxygen attached to it, right? Valine is another one, right? Only 3 carbons. So some are large, some are small. You'll also notice that some of them might be polar, okay? Can you figure out which ones would be polar? If you're thinking the ones that have oxygens and nitrogens, you're correct. Anything that's got these oxygens on them, like glutamic acid, tyrosine, threonine, serine, and arginine, these are reactive. They can react with water. They can react with other polar molecules. You see this valine, how it's just all carbons, and the phenylalanine is all carbons? Those are nonpolar. Those are hydrophobic. Okay, so they're going to generally repel water, and they're going to be attracted to other hydrophobic side chains. Okay, now if we take our amino acid chain and we simplify it even more, we can see that we just form the backbone, okay, which shows us just the sequence of amino acids. And in our backbone, we can have our side chains alternating up and down, up and down. But to even simplify it so it's easier to read, we can just assume that our backbone is linear and rigid, and you've got all the side chains on one side. And that's just so we can make sure we can see what everything looks like. Okay. This is known as the primary structure of a protein. It's simply the linear chain of amino acids. Okay, and a primary structure is the first structure that a protein forms. So all it is a linear chain of amino acids. It's a polypeptide chain. That's it. The sequence of amino acids, it's not random. Always remember that. It's determined by the DNA. So the sequence of bases is going to determine what the amino acid sequence looks like. And the sequence of amino acids is going to determine how a protein folds. So if you change one of the amino acids, you're going to change the structure of your protein. And if you change the structure, you change the function. Okay? So the sequence is really important because that R group amino acid sequence, the sequence of those R groups is going to determine how a protein folds. And one of the videos, it's the second video link on YouTube, explains how proteins fold. It talks about the secondary structure of proteins, and then the last video, the third one, gets into that folding in more detail. It's all about those side chain interactions. Okay, so these are what the side chains look like. Now, the one class did this, the other class didn't, thanks to COVID. So we weren't able to do this as a group. But basically, this diagram represents the 20 amino acids that form proteins. Okay. And these little models basically show the structure, and the colors tell us whether they are hydrophobic or hydrophilic, if they're polar or nonpolar. So basically, what we did is we modeled proteins with tubers in one class, and we put the side chains together. There are 3 basic principles of chemistry that deal with bonding. The hydrophobic amino acids always tend to cluster in the middle, where there's less water interaction. The hydrophilic ones like to be on the outside because proteins exist in a watery environment, And so anything that's hydrophilic, that's charged or polar, is attracted to the water. Um, Opposite charges will always attract and form a salt bridge. So those would be the highly charged amino acids, okay, and they're usually colored red and blue. And then cysteine is a unique amino acid. What that does is that forms a disulfide bond, okay. And so the thing to remember is that the shape of a protein is predetermined by the amino acid sequence. Not all amino acid sequences are functional. So if you just took a random bunch of amino acids and put them together, it may not form a functional protein. It might just make a glob, and that's it. A sequence could result in a terribly shaped protein. So what's important is stability. So when proteins were first forming, not all of them came together and formed nice structures. Some of them were a horrible thing that did nothing and it fell apart. So the amino acid sequence really is important in determining that. Okay. All right, let's get to secondary structure. Okay. So when a polypeptide is built, it will begin to coil and form. And the reason it does that is because of hydrogen bonds in the backbone. And it forms one of two possible structures. One of those structures is called an alpha helix, and an alpha helix basically forms a coil. Okay, alpha helices are represented in 3D computer models like this, where you see this corkscrew turn. Okay, the parts in between where you just see this little loop, that's known as a turn. That's it. So the side chains, they can play a role in whether the shape forms, but what really determines the formation of the alpha helix is the hydrogen bonding that goes on. Now, alpha helices, when they form, the side chains generally stick out from the sides. Looks like branches of a tree coming out. In a beta sheet, the side chains interact with each other, and they generally stick out along one side or another. And with a beta sheet, what you get is this alternating, it looks like an accordion, it looks like a folding pattern. And a beta sheet, the hydrogen bonds, they form laterally or sideways, okay? And so a beta sheet is usually represented by a flat ribbon in a 3D model. And you'll see this when you get into JMOL. Okay, so we have alpha helix and beta sheet. Now, if you put these together, they can happen at the same time in a protein. You get what's called tertiary structure. Okay. Now, the things that hold proteins together, there are four main interactions that hold them together. There are van der Waals attractions, where the hydrophobic nonpolar side chains often bury themselves in the center of the protein. We have disulfide bridges, which can hold the two cysteines together. We also have hydrogen bonding, which plays a huge role in the formation of alpha helices and beta sheets. And then there are also ionic bonds, right, which can form between oppositely charged ions that are part of side chains. Okay, when these come together, you get tertiary and quaternary structures. Okay, and a quaternary structure is the final maximum three-dimensional configuration of a complete protein. Okay, and so what you wind up with is this is an antibody. And it's kind of interesting. This is one of the proteins we talk about. They're looking at antibodies to treat people with COVID infections and extracting them from the blood. Antibodies are proteins. They're Y-shaped proteins, so it's kind of has a little bit of a Y shape here. And it's made up of several chains. It's made up of light chains and heavy chains. And so you actually have a total of 4 chains. You have 2 light chains and 2 heavy chains. So it's a complex protein, but it's called quaternary because it's made up of at least 2 or more polypeptide chains that come together. Okay, and you can see in here, if you look, you can see where there are beta sheets, right? They're indicated by these flat ribbons, and you can see some of the alpha helices. If you look at the red structure towards the bottom, here's a loop, right, and here's a loop. Those are your alpha helices, right? And there's another one in here too. Okay, that's quaternary structure. Okay, here's another one. This is hemoglobin. This is what hemoglobin actually looks like. It's loaded with alpha helices. Okay, you see all those helices? See how they're coiling? Okay, and so you've got 4 parts to this. Okay, you've got 2 beta chains, which are colored yellow, and 2 alpha chains, which are colored blue. The quaternary structure also has what's known as a prosthetic group in it. There are 4 heme molecules in there. The heme molecule contains iron, And the role of the heme molecule is to bind with oxygen. Okay, so the iron that's in our blood is actually embedded within the hemoglobin molecule, and each hemoglobin molecule contains 4 iron atoms, or heme groups that contain iron atoms that carry the oxygen. The iron in the heme group is what binds the oxygen so we can carry the oxygen in our blood. Okay, and the hemoglobin is made up of 4 separate protein components, 4 polypeptide chains. That's why it's called a quaternary structure. Okay, let's see if we want to look at these. Really, probably the biggest thing we'll talk about is a domain, which is a part of the protein that has a specific function, right? That would be like an active site, or it could be an area where a different molecule can attach to it. Lobes basically are just areas that have a specific chemical nature. For example, there are hydrophobic lobes and hydrophilic lobes. There might be an ionic reactive lobe. Okay, so those are just some of the important terms that you might come across when we're talking about proteins. Mostly domain and lobe come up. Okay, a cleft, it's basically a cleft. It's like a space or a ravine or a valley. It's an area between proteins. It might be a regulatory site, these clefts, and a face is usually a flat surface of a protein that has lots of beta sheets. Okay, so how do we look at proteins? Well, the first thing we have to do is you have to crystallize your protein. And actually, before you do that, you have to isolate your protein. So you isolate a protein, and there are many ways to do this. I've never isolated them, but it takes a long time, and it takes um, a lot of different types of compounds to do so. But once you isolate a protein, you can crystallize it. You can force proteins to grow and form crystals by placing them in different types of conditions. It's basically trial and error. Scientists try different types of combinations of compounds to force proteins to form a crystal. Once you have a crystal of a protein, that crystal consists of millions of repeating subunits that each have an individual structure of the protein in it. And so what we can do with those crystals is we can bombard them with X-rays and we get a diffraction pattern. Now that diffraction pattern contains essentially the marks of electrons. It shows where the electrons are in that particular protein crystal. From that diffraction pattern, computer programs that have been developed can read the pattern and create what's called an electron density map. It shows us a map of where the electrons are located. And the area where the electrons are the most concentrated, that tells us where, oh, there's a carbon, here's a nitrogen. Oh, this looks like a side chain of arginine or threonine or phenylalanine. And then from that map, we can fit that using programs, which can help us develop an atomic model of the protein. Okay? And we can resolve the protein. So basically, if you look at a protein at 5 angstroms of resolution, you can see that you don't really have a good picture of the protein. But as it gets resolved down to a lower resolution, which means you have a smaller number, okay, now we get a much better picture of where the different atoms are located in the protein. So you see how these electron clouds, represented by these squiggly lines, tend to fit the shape better, and we see that there's a much better shape to this. Okay, that's a better resolution. Okay, at 5 angstroms of resolution, it's pretty much like a column with a couple of little blobs hanging off. But as we go down to 3 and then 1.5 angstroms, we get a much better, closer look at it. It's kind of like a microscope. When you're under high power, you get to look at things in greater detail. When you're under low power, you tend to see more of an overview. And that's basically what's happening here. 5 angstroms is as if you're under low power of the microscope, so you're seeing a bigger picture. And then as we zoom in to 1.5 angstroms, we get to hone in and target and pinpoint specific atoms. Okay, so that's how we resolve it. These are just some of the common protein motifs. We get what's called a sheet helix sheet. Um, Sometimes you get a helix turn helix, and so those are just some of the different motifs. We really come across these when we get further into protein structure. And then the next piece of this will be protein crystallography, which I will talk about in another video. So at this point, you should be pretty well caught up to this point of the notes. And then moving forward, we'll get into protein crystallography in the next session. So I hope that helped you guys out. I hope you got a little bit of a better idea of this. And moving forward, I'll post a video about crystallography, and that will allow you to understand a little bit more of how we do the work we do at Brookhaven Lab. And for those of you that are interested in doing this work at BNL, this is what we'll be doing more often. So I hope that this was helpful to you guys. Stay healthy. I'll see you soon.